arrow at the present now, isn't it? That I have put your entire screen or a window, should I say?
You can go with screen, sir.
Okay, okay. Application, share an application window. It's not coming actually. What is this? Again, it got muted. No account required. All these things, there are some pop-ups, but I'm not able to. Hello? You're able to see my PPT,
No, sir, we can only see you, sir.
but the sharing is not coming, isn't it?
I have sent you, sir, how to do it. I have sent you the screenshot once you check on your WhatsApp.
Yeah, I have. See, whenever I am asking share, it is coming. Share is blocked. Share is blocked here. You are able to see my screen?
No, sir, we can only see your face like on camera.
That is exactly when I am hitting the share window. You are able to. You are able to hear me.
Yes, sir, we can hear you.
You can also. You can view this particular PPT also. I think the my figure may not be visible. Stop sharing. Ignore. Okay, Google.com is sharing a window. Okay,
Now we can see your screen, sir. We can see the presentation.
I can start presentation.
Yes, sir, you can start now.
Okay. So this is actually the model. This is the present model of glycolysis. Okay. I told you about the top-down model and also bottom-up model. I think who is that new girl? She was not there, but let her understand this thing that this present model on the left side, extreme left side, is what is called a top-down model. According to this model, glucose is phosphorylated in two-step reactions to fructose 1,6-bisphosphate, and then it is split into triose phosphate. It goes down into these things. There are several constituents of this model. This model represents apply in biology even to this day also. One of the major problem that is actually one of the major problem is this model considers it as what is called respiration is a catabolic process. Glucose is catabolized to pyruvate in cytoplasm. It produces ATP in cytoplasm at the phosphokinase level and glyceraldehyde with 1,3-bisphosphate, that is PGK1, phosphoglycerate kinase. Okay, and then the debate on the role of carbon dioxide, oxygen, alcohol, lactic acid, citric acid metabolites remains almost for 100 years. So there are several confusions on that. My attempt is to resolve those confusions. Now, despite actually wonderful data that we present in actually cell work systems or animal models, we are not in a position to actually find proper therapies for diabetes, cancer, metabolic disorders, viral infections, blah, blah, blah. So in this particular case, what could be the reason? My assumption is was actually my assumption was now I am very clear that this model needs a thorough revision. Unless this model is thoroughly revised, we will not be in a position to actually develop proper therapies is this thing. So our attempt is actually to remodel these things and then how these models were developed. Before that, let us understand some of the basic principles of actually maintaining health. Okay, it started with Claude Bernard, who has introduced the concept of homeostasis and constancy of internal environment. He has also stated that nutrients are actually converted into glycogen like materials in the liver, and liver supplies sugar to the blood. Walter Cannon, are you able to hear me? Hello?
Yes, sir, we can hear you.
Oh, okay, even if you don't, are you able to see me?
Yes, sir, we can.
I am not able to see anyone. I am only seeing on this thing. As long as you are able to hear me and see the screen, that will be wonderful. Okay?
Yes, sir. Yes, sir.
Okay?
We can see the screen. We can even hear you.
Yeah. See, there are actually three developments that have actually played a critical role in developing actually the present models of metabolism. Okay, apart from Claude Bernard's glycogen theory in case of liver and supply of glucose from the glycogen by liver, there were other cases like what is called Walter Cannon stated that feedback loops control actually the metabolism. Second important thing which we have come across is defect in pancreatic islet secretions cause diabetes, right? Banting and McLeod purified insulin in 1922, and January they gave it to the first patient, Leonard Thompson. Okay, and this patient, although he died in the early stages, but subsequently, actually, another person lived up to the age of 90 years when insulin injection was there. Schoenheimer introduced the concept that there is a dynamic state of the body, and this dynamic state of the body means every tissue has a turnover. Excuse me. Every tissue has a turnover, and this turnover is maintained by macromolecular recycling. That is, nutrients are taken through the diet, digested, incorporated into the macromolecules through the metabolism. So there is a turnover of macromolecules to monomer units. Monomer units are rebuilt back into the macromolecules for the fundamental concept. The present model ignores that particular concept also. Then Schoenheimer used actually the radio labels to demonstrate that thing. Then cell culture techniques came into picture at the end of the 20th century. There was a hype that cell culture can be used for regenerative medicine. But even to this day, we are not in a position to optimize. That I again say is because of the defects in the present models of metabolism. Another important concept is Weber introduced the concept of symmorphosis, where he has suggested that there is a perfect economy between the structure of tissues and their functions. So, microenvironment, structure of the functions, structure, and then the dynamic state, they need to be coordinating actually these things. Okay? Now, you look to anything, if you look casually, any paper also, whether it is neurodegenerative disorder, diabetes, or carcinogenesis, or infection because of the viruses or pathology, you will understand that epithelial cells have to undergo modification into a mesenchymal state. mesenchymal state. In stem cell cultures, we will also call it as a pluripotent stem cell. There are two types of stem cells. So one is totipotent. A zygote is a totipotent cell. It produces multiple cell lines. But whereas a pluripotent cell, there is a lineage fixation where actually a particular cell becomes will be modeled, remodeling its metabolism for a specific type of cell. In turn, in cooperation with other cells in the microenvironment, together they constitute what is called a tissue. So, tissue dynamic state, in the first stage, tissue is undergoing actually degeneration and then it is rebuilt in this process. This is how actually health is maintained. Now, I'll come to this particular things a little later again, but my attempt here in this particular case is any cell that is actually rebuilding its metabolism is to re-enter into the cell cycle, and this cell cycle has to be completed to make actually a perfect cell rebuilding into the tissue. And in pathology, that is what is deregulated. That is what Sheffield has stated, that it goes on actually increasing the proliferation without any limitation, constraint in case of pathology. In case of diabetes and other neurodegenerative disorders, the cell cycle is incomplete and cell death is activated in these things. Okay? Are you able to, have you understood? Hello?
Yes, sir.
Yes, sir.
Shall I
Yes,
move
sir.
on to the next? Okay. In the very beginning, I told you that this model talks about what is called this particular respiratory catabolic process. Okay. This particular thing is based on what is called that simorphosis or dynamic state of the body were not analyzed. Second important thing is there are certain changes actually that have taken place. I think I will come to this particular slide later. Okay. Okay. Now, how actually the present models developed? Number one, I don't know why this is coming. I'll just 1 minute. I'll get back to you again.
Okay, sir.
You are able to. Okay. We'll start with this. Centuries of research on muscle contraction, alcohol, and respiration are the basis of this thing. In addition to this, you can add endocrinology, hormones, and other things. Okay. Aristotle introduced actually that there is a spirit pneuma, which is called a vital air, that actually increases the breadth of the muscle and reduces length of the contract. That means it is a respiratory process that plays a critical role in this thing was known. But primarily, he recognized the fact that exercise and physical activity in muscle, that is keeping actually the health. That is what your yoga and then physical exercise is what is actually doing. And then when it came to muscles, Galen introduced the concept that muscle is made up of fibers and flesh. Myosin portion is considered to be the flesh portion, and fibers are continued into tendons, which attach actually the muscles into the skeleton. Okay. In 17th and 18th centuries, alcohol definition and also muscle definitions came into picture. I have given you this thing. This slide will be there in front of you. Therefore, you can make notes out of it. Now, let us get into this thing. One is alcohol. First, alcohol was defined as a fermentation of a violent internal motion of fermenting sugar in an aqueous environment where alcohol remains in a liquid form, and gases like carbon dioxide will be evolved, was given by phlogistic theories. Lavoisier, in fact, who has overthrown actually the phlogistic theories, has sustained this thing. He has also stated that both carbohydrate and then alcohol is made up of carbon, hydrogen, and oxygen. Okay, Lavoisier also talked about oxygen, but Lavoisier's concept of oxygen is different from the present model of concept. I'll come to that later. But 19th century experiments on muscle contraction, which focus mainly on lactic acid, okay, that in 1795 isolated frog muscles maintain irritability was shown by these people, and then Scheele has identified lactic acid in these things. Okay, now let me give you some of the important points. Anaerobic fermentation, Pasteur's experiments in 1850s, right? How they have defined fermentation, how they have defined actually the growth of yeast. Pasteur has introduced the concept of aerobic and anaerobic life. That is, anaerobic life is life without oxygen, and then aerobic life. This particular slide you carefully study, because it gives you actually the summary of our model also, which was distorted by actually the Warburg and Meyerhoff in the early stages. Okay, when a local producer of alcohol from beet sugar, he used beet sugar, that also you remember, right? Monsieur Bigot, he has serious problem in fermentation industry where yeast was growing like forest, but alcohol was not produced. Now, Pasteur observed the yeast that were there in this particular industry under the microscope. Under the microscope, he has seen that when alcohol is actively produced, the yeast cells assume a globular structure. But when they are growing, they become what is called lengthened, and then they assume the filamentous structure. Do you understand? This is important point. Hello? This is important point. That is what we are talking about EMT, epithelial mesenchymal transition. Epithelial transition is epithelial cells are actually polarized cells attached to other cells in their neighborhood. But mesenchymal cells are isolated cells living in the microenvironment independent of polarized tissue. But they live in the company of other type of cells in case of eukaryotes. Have you understood this thing? This was recognized by Pasteur in those days only. But he has assumed that the structure changes during the fermentation process. It assumes globular structure, but when it is growing again, it assumes its filamentous structure. Is it clear? Is it clear?
Yes,
On the
sir.
growth of yeast, he has he has actually placed the yeast in ammonium tartarate solution, and even there also he has seen that there is a simultaneous alcohol production and the growth also. But depending upon what is called the amount of the lactic acid that is produced, alcohol production will be suppressed, growth will be activated, and during the growth, nitrogen is incorporated into a protein material. He called it as albuminoid-like material. Okay? Did you understand the difference?
Yes, sir.
Yes, that is, there is a simultaneous there is a simultaneous fermentation process and also a growth process. That is biogenic process. Is it clear?
Yes, sir.
Growth is biogenesis. Fermentation is breakdown of actually nutrients into the fermenting material like what is called alcohol. And when lactic acid is produced, he has stated that lactic acid contamination suppresses fermentation and then promotes growth. During the growth, it converts nitrogen to the protein. Okay? Then he has also stated earlier to that Liebig thought that it is a fermentation, is a putrefaction process. He didn't, and he thought that oxygen rescues actually the yeast or organisms from the, or yeast are produced because of oxygen in that fermentation process. That was his theory. Okay, but this was contested by Pasteur, and Pasteur has stated clearly that fermentation is a living process. It is anaerobic process. Now, by that time, enzymes were not popularly known, but pepsin was known. And somebody during that period suggested that, yeast may be actually containing pepsin-like material, which will be converting glucose or sugar molecule. It is not glucose or fructose, they use that term. Sugar was used amorphously. This sugar molecule will be converted into alcohol by that particular enzymatic process. On this, Pasteur was very categorical that he is not, unless he investigates personally, knows about it in detail, he will not be in a position to comment on that thing. Therefore, he said that, either neither accept it nor deny it, but should we say that something in the yeast in a form of fermentation, in a form of enzyme, will be converting actually sugar to glucose, or yeast itself will be converting it to yeast itself will be converting sugar to alcohol? That was the question which actually he did not touch. That is the honesty of Pasteur. Okay, Pasteur did not work on enzymology. It was only Buchner who in the in 2000, okay, at the turn of the century, between 1980 to 2000, he has actually introduced, no, sorry, it is 1890 to 1900, he has introduced actually the cell-free extracts. See, this Liebig Pasteur controversy was examined by several people as a process of that thing. What Buchner did is he crushed the yeast cells, extracted the dry yeast, and that he actually dissolved and then saw whether it can produce alcohol by adding sugar molecule. In that, he was able to demonstrate that cell-free extracts have the ability to produce the alcohol. Okay. Now this was extrapolated by the central figure here Harden and his student Young to produce what is called alcohol production and optimize the conditions under which alcohol production is there. Now, Harden contributed to two things here. I'll come to that later, but let me examine that, what actually Pasteur demonstrated is under anaerobic condition, sugar 60 grams per gram of yeast will be consumed, but in the presence of oxygen, only 4 grams per gram is utilized. Now, lactic acid suspends fermentation, phosphate acts as a mineral. He did not know the role of actually phosphate. Nitrogen promotes actually biomolecular synthesis, that is albuminoid-like material. Okay, so growth is always associated with nitrogen utilization was clearly demonstrated by Pasteur. Okay, contrary to that, we still upon what is called, depend upon what is the carbohydrate metabolism alone. Okay. Now coming to the Harden and Young, in this, his student worked extensively on that. Harden was a senior fellow who has actually initiated this particular work and then provided the facilities. Under that, he has identified that, if the yeast is extracted under the cold condition, the production of alcohol was very less. Then, by coincidence, he added what happened. Part of the yeast juice boiled. He boiled the yeast juice and added that particular hot portion to this particular yeast extract. Then suddenly the alcohol production boosted. So in that case, Harden and Young in 1905 came up with a theory that there is a thermostable cofactor which is essential for fermentation process. If you limit sugar or if you limit phosphate, fermentation will not take place, but fermentation, whether it is activated by phosphate or sugar, it requires actually these thermostable cofactors are known to that. Along with Harden and Young, others also worked. I'll come to that in the next slides. Now, what Warburg's role in this particular thing? Warburg introduced two concepts. One is posture effect. He stated oxygen suppresses fermentation. That was not the theory which Pasteur proposed. It was lactic acid which suppressed fermentation. Second important thing, he introduced aerobic glycolysis. Under that, he has stated that the glucose is actually converted to pyruvate in cytoplasm, and pyruvate is actually converted to carbon dioxide in mitochondria in the presence of oxygen, but in the absence of oxygen or in the defective functional mitochondria, there what happens is pyruvate is reduced to lactic acid. That is what we call it as a this thing. Now, I'll explain you this particular concept in a more detailed manner. He introduced Warburg experiment and other things where signal cells were taken and their respiratory quotient. Now, as per yeast is concerned, respiratory quotient in that Warburg experiment is okay, but applying or extrapolating that to the human tissues is one of the blunder that was committed in the first quarter of the 20th century. Okay, now Warburg's focus was only on ATP production. ATP was not discovered up to 1927. 1927, first actually Fiske and Subbarao discovered ATP, and Eggleton discovered creatine phosphate. And these two are important molecules came to this picture, but focus remained only on ATP and creatine phosphate. That is one another bigger blunder of this present models of metabolism. And Warburg was keen on that thing. In his 1956 paper, The Origin of Cancer Cell, which is considered to be the hallmark of cancers even to this day, he says that any other explanation other than ATP production and lactic acid production will be delaying the disease, but despite 100 years of Warburg effect, we are not able to cure the disease. Okay, now this is, have you understood this one? Okay, let me go to this thing. What did Lavoisier say? See, these people are only in the neighborhood, German, France, and other things, they would have exchanged easily, understood actually the works done by these people clearly. But Lavoisier's work was published later by his wife, maybe they miss some of the important things also. But we need to understand, even now also we are harping on Warburg effect. Therefore, we need to know what is the function of oxygen inside the body. Right? Now, Lavoisier defined oxygen as burning of fuels inside the body. Only that statement is taken to state that present models of glycolysis carbohydrate is reduced to carbon dioxide. Lavoisier did not talk about protein nor on fatty acids. Its concentration was only on carbon, hydrogen, and oxygen. Lavoisier never used the term carbon dioxide. He only used the term carbonic acid. And he stated that this carbonic acid is produced during the digestion and utilization in the tissues, and this is exchanged with the atmospheric oxygen in the lungs. In addition to defining this, Lavoisier also has stated, experimented with shrews and pigeons, and stated that oxygen increases the weight of tissues during the growth of the organism. If the respiration is suspended, the weight of the tissues or growth of the tissues will not take place and the animals will die. Okay? Then, on the toxic effects of oxygen, Lavoisier also has recognized that oxygen is toxic, and this toxicity causes actually production of noxic hydrocarbons. And these noxic hydrocarbons are excreted or removed from the body in the form of sweat through the skin or in the form of what is called evaporated vapors from the lungs. In winter season, when you blow air from the lungs, you will see that it goes like vapors, right? So that is nothing but a carbonic acid vapors that will be going out, which are thrown out of the body, which if they are retained inside the body, they remain toxic also. So this dynamic state of the body, what it is doing is, it is taking out fresh nutrients from the food, incorporating them into the tissue, and in the whole body, this tissue, after the tissue metabolites are burnt, converted to carbonic acid, they are thrown out through the lungs. Okay, that is the summary of this thing. But Lavoisier never discussed the source of carbonic acids, nor the source of nor proteins, nor fatty acids to that matter, because at that time it was not understood fully in that thing. Okay. Scheele isolated actually lactic acid in the sour milk in 18th century along with the with Lavoisier. There is also a complaint that Scheele also discovered oxygen, but that is for litigation people. Let us not get into that thing. But lactic acid became important with one simple observation of Berzelius. Berzelius was considered to be one of the father of chemistry. He introduced actually many of the chemical concepts in that particular 19th century. So Berzelius, in the first decade of 20th century, he has reported that the hunted stags, which are used for actually for eating, that is for cutting the muscle, they produce more lactic acid when compared to the resting deer, which are slaughtered for extracting the muscle. That means there is a variation between the exercise of the muscle and resting muscle. The exercise muscle, which is dying, in death, after death, it has a lot of lactic acid was Berzelius. Berzelius did not discuss the dynamics of lactic acid production nor accumulation. He only stated a fact that when hunter stags are killed, their muscles contain enormous amounts of lactic acid when compared to the resting diet that were considered. It was subsequently what happened is there were several people, Leibig and others, who have actually reported that the lactic acid also circulates in the patients suffering from leukemia and other pathological disorders. But it does not accumulate, it accumulates only in dead bodies. That means lactic acid is a dynamic molecule which is produced and also consumed during living process. Have you understood? This is an important point which you need to note from the 19th century studies. That is, lactic acid is produced, no doubt, in the living conditions, but it is consumed during the living conditions. But when the organism is dying, lactic acid accumulates in their tissues. Is it clear?
Yes, sir.
Now, this particular thing is ignored by again by carcinogenesis. What is carcinogenesis? It is increase in cell number. Increase in cell number means it
Cancer,
is a
sir.
biogenic process. It is a biogenic process, biosynthetic process. It is an active living process. Then there's a hyperactivity in living activity in production of biogenic materials. Isn't it? Right? Have you understood? Have
Yes.
you understood the reference? What they have stated is during the living condition, lactic acid is circulating in the blood but does not accumulate in the tissues. But in dead bodies, they find actually lactic acid. The stiffness you will see that in the if supposing some patient is dying, the muscles are actually twisted like this thing for some time because they should not become stiff. That stiffness, rigor mortis, we call it. That rigor mortis develops because of the accumulation of lactic acid. Fletcher in 1869 started his work. Before that, actually, there was another person whom I have already mentioned in 18th century, who has demonstrated that if a slaughtered muscle is kept in an oxygen environment, it maintains its freshness. But that is for some time. But when the oxygen is not supplied, it becomes stiff. But he has also demonstrated that there is a variation between the oxygen requirements at high altitudes and at normal living level. If a man is going from the normal plains to the Everest, he requires oxygen equipment as a supplement for oxygen. So, oxygen supplementation over a particular partial pressure of the oxygen helps in actually survival of the organism was shown on that thing. Fletcher worked on that. Fletcher, what he did is he isolated a frog muscle, optimized the conditions of actually maintaining that thing in the experimental models, and there he has also optimized the estimation of lactic acid and carbon dioxide methods. And then he has stated in 1869 paper, it runs around 300 pages. So under that particular thing, he has stated that several experiments, each experiment details he gives. Under that, he has explained that as long as the oxygen supply is there, muscles will maintain the carbon dioxide production, and lactic acid, he did not find it during the particular process. But when rigor mortis developed, he has stated that there is a limitation of life in the isolated muscle. In the isolated muscle, or even if the oxygen is supplied after certain time, the muscle develops into rigor, that is, slowly it dies. That death results in the accumulation of lactic acid. So rigor mortis develops actually lactic acid was shown in that particular period. Okay, have you understood that? Hello, Janavi,
Yes.
you have understood?
Yes,
Yes.
yes, sir.
Oxygen supply to the to the isolated muscles has its own limitation was understood by Fletcher. In 1907, he also gave his article Hopkins as a co-author, but surprisingly, later on, Hopkins delivered lecture. Actually, this Croonian lecture was delivered in 1909. And by that time, actually, Fletcher went into what is called, left Cambridge, and then 1911 or so, Fletcher expired. I don't know whether Fletcher had access to this particular paper, which was read by Hopkins or not. My hunch is whatever actually Fletcher has given in summary in 1907 was actually reversed by Hopkins. Hopkins depended on what is Hill's work on heat production, thermogenesis, that was taking place between the first decade of 20th century. Both Meyerhoff and Hill shared 1922 Nobel Prize. Okay, I'll come to that little later. But did you understand the difference between? Now, I'll discuss a little more on 1907 review. You can also look to that paper. All these papers are easily available. Okay. What actually Fletcher, during that particular period, two things happened parallelly in health of individuals, that is diabetic related individual research. Number one is this man has discovered that what was his name, Bernard, right? Bernard discovered actually the liver is the central stage where glycogen is produced, and then it also secretes lactic acid. Then, after two decades or so, Minkowski stated that no, islets of Langerhans secrete a thing which actually maintains the blood sugar levels. So if you remove the islets, you will not be in a position to actually control the diabetes, was his theory. Now there are two uh, three principal debates in 19th century. I'll not go into the details, but I'll give you summary of it. Number one, Minkowski's argument that only islets are responsible for maintaining blood sugar, although was published by Flüger Archius. Flüger was a Nobel laureate by that time, and then he maintained that journal. But Flüger contested the theory of islets contributing to this. And that confusion even now exists. So I'll come to that part later. What Flüger has stated is it is the nerve and blood vessels which are actually supplying from the connecting duodenum neural system to vascular system to the pancreas and the liver organ systems inside the body, inside the abdominal region. They have a control on actually maintaining the blood sugar level was there. But unfortunately for him, what happened is McLeod and Banting, somebody in between actually by ligating that particular pancreatic duct, first thing that happens is exocrine glands of the pancreas will die. Endocrine glands survive for longer time, and those endocrine glands can be isolated was shown by Banting, and MacLeod and Banting purified that and then started this thing. Okay, I'll come to that later. There are fallacies in that also. We'll come to that little later. Have you understood? The second important problem was Du Bois Raymond introduced the concept of galvanometer recordings, stating that tissues contain what is called electric potential, and this electric potential muscles have a natural what is called electric potential was what was actually the argument of the Bois Simon. That is what we call it as a resting potential and action potential in case of nerves. Okay, his student Herman, who worked with him, differed with him enormously and has stated that some injury and loss of actually an unknown molecule called as ionogen was responsible for this particular change in the electric field. Surface of the muscle is electrically neutral if it is healthy, but if there is a injury to the health of the muscle, then there will be what is called electrical differences between the interior and the exterior of the muscle was what the Herman's argument. There was a debate and that led to the personal level. Even Buchner, even Minkowski's and Flugers also left it to this particular level. And you have actually Fletcher and Hopkins, where actually lactic acid role and thermogenesis role also came into this picture. These are some of the major things which you should keep in mind when you are doing research, and in that when you are trying to get a wet lab data, keep these things in your mind. Okay. Now, these are the architects. The first three are the architects of the present models. In 1920s, Nobel Prize was given to both Hill and this thing. What Meyerhoff stated is glycogen is converted to lactic acid and that produces the energy. He introduced the concept of certain fluxes, and this fluxes will result in the production of the lactic acid, and that lactic acid production supplies the energy to the contracting muscle. In other words, he introduced a very technical term which is known as anaerobic capacity of the muscle. Anaerobic capacity of the muscle. That is actually another form of the aerobic glycolysis or posture effect. Okay, but they didn't talk about posture effect or aerobic glycolysis. During that period, in parallel in Cambridge, Meyerhoff was working in Germany. Hill was working in Cambridge, and in Cambridge, he observed that whenever there is temperature increase, when you put actually the muscle on an experimental tool, it increases the temperature. When you run also, there is an increase in temperature. Hill is considered to be the pioneer in the exercise physiology also, and that thermogenesis results in the accumulation of lactic acid, and then after the exercise, the muscle or the body takes excess oxygen, and this oxygen he called it as, because we gasp it when we run and then stop running, you take more of oxygen inside, isn't it? That he called it as oxygen debt. This oxygen debt, he stated that part of the lactic acid is reoxidized to supply energy, and that energy is converted to glycogen. Now, that energy is used to convert lactic acid to glycogen. See the contradiction there also. Glycogen conversion to lactic acid produces energy. Part of the lactic acid is oxidized to utilize the energy, to produce glycogen. That means gluconeogenic processes require actually part of the lactic acid was recognized by him, but the interpretation was lactic anaerobic capacity was the interpretation. I'm telling you these two things very emphatically for two simple reasons. Glycogen metabolism is independent of lactic acid metabolism. Have you understood that?
Yes, sir.
So that is exactly the error that exists in the present models. Up to 1934, 1940s, Meyerhoff actually promoted. Actually, in 1927, Fiske Subbarao and Eggleton got discovered creatine phosphate and other things. But Lohmann, who was working actually with Meyerhoff, was working on several other things also. He was given Nobel Prize in 1929. And in that, after that, getting the Nobel Prize, Meyerhoff and Lohmann worked together again and they discovered the phosphoenol pyruvate. Lohmann was not involved in another theories. Lohmann discovered only in this thing, 1934, phosphoenol pyruvate. In 1938, by that time, Nigerian from the Warburg group has introduced this particular concept that that glyceraldehyde 3 phosphate and other things will be converting into pyruvic acid. So they went by that particular thing, and but after 1945, Meyerhoff drastically changed that thing. Lardy and Ziegler in 1945 reported that pyruvate kinase produces phosphoenol pyruvate but not pyruvate. Second important thing is Meyerhoff we revisited Harden and Young equation in 1945 and discovered that there is an enzyme called apyrase which hydrolyzes the ATP. That is, ATP produced is hydrolyzed if it will not be allowed. So, ATP is either exchanging phosphate between the organic molecules or it is hydrolyzed. But this hydrolysis takes place by an enzyme called apyrase, and this apyrase takes the ATP to the exterior of the cells. There in the microenvironment, it attaches to what is called the extracellular membranes of other cells. There they will be hydrolyzed. The ATP is the enzyme that will be hydrolyzing. We'll come to that later. And in 1949, it took 4 years after Ziegler has reported that phosphoenol pyruvate is produced by pyruvate kinase but not pyruvate. Then he reinvestigated the equilibria. Remember, all these experiments were done on a single step reaction using enzymes, isolated enzymes, and adding substrates. And when the reaction is reversible, what you add becomes important in catalytic reactions. Do you understand that? So, if you add phosphoenol pyruvate more, it may produce pyruvate. Or if you add pyruvate more, it may produce phosphoenol pyruvate. But in pyruvate kinase case, even that is also not there. Pyruvate kinase is a reversible enzyme, was the assumption made by this thing. These papers are somehow not published in textbooks or nor discussed in these things. There is one paper on the 100th centenary years of JBC, Journal of Biological Chemistry, which contributed enormously to the development of biological chemistry or present biochemistry. You can note down that Kresge 2005, Kresge et al. There are three authors, I believe. Kresge 2005, Otto Meyerhoff, and origin of glycolytic theories. Okay, if you hit it Google, you get that paper. In that paper, you find references. And another paper, and actually, I stumbled on that paper that in 1980, Racker, who also worked along with these people extensively independently in USA, there he wrote a review article from Paschal to Mitchell. Mitchell was the one who has introduced the concept of complex 5 and also this potential, right? Complex 5 and then membrane potential. Change in the membrane potential that creates ATP is the Mitchell's concept. Now, in that paper, he mentioned this 1945 paper and made a note also. Somehow this paper is not published, not either it is not allowed to be published, and many of the people are not aware of this paper, and therefore they are ignoring this particular concept. Mayer have altered his opinion on this thing. Okay, now coming to Hill. Hill in 1920s stated that lactic acid actually reoxidation produces this thing. But during 1925, takes in Australia, he has actually reported that by the time we have understood creatine phosphate came into this picture 1927 or so. So during that particular period between 1925 and 1940s, takes published few articles. Under that article, he has clearly stated that the creatine content will be lost during exercise after the exercise when the rigor mortis develops. But when the tissue is respiring, the creatine is retained inside the tissue. That means there is a dynamic state of production of creatine was recognized by takes. Okay, because of the limitation of experiments and experimental models and equipment also, they couldn't go beyond that. But I am highlighting actually the concept which actually takes has created is during respiration there is a process of actually keeping creatine phosphate constant in the muscle, and creatine exchanges its phosphate with ATP, and then this creatine phosphate is produced. Creatine phosphate is stored, ATP is never stored inside the cells. That is why, even to this day, we do not have an optimal levels of ATP that are produced in any tissues. Okay, it is only either in the form of actually phosphate exchanges through magnesium ATPase or through creatine phosphate production in excitable tissues like muscles and nerves. We estimate ATP content in the tissue. Uh, And in 1932, there was after publication of Take's paper, there were also other papers. During that period, some gentlemen commented humorously on what is called these isolated tissues and then science. He has commented that when you are using isolated tissues and extrapolating those results to living bodies, these errors are bound to come. And to this, Hill acknowledged the humor in that and also the satire in that, but he was noble enough to concede that science is a growing subject. At one particular time, we work on particular concept. This concept is re-examined, and examining during the examination, we come to know about what is called different models. Either we alter the model or we improve the model, amend the model, explain unexplained issues in that. That was the lecture given in 1930s that was published in American Journal of Physiology in 1932. under the title of Revolutions in Muscle Physiology. What Hill stated is discovery of creatine phosphate is a revolution in the muscle physiology. What he has stated is creatine phosphate is utilized during the contraction for supplying the energy, but after during the rigor mortis, creatine is excreted as a degraded form in creatinine form into the urine that will be excreted into the blood. From there, it goes to the urine. So it has to be resynthesized, right? So he considered that thing, and that particular thing he has assumed that creatine phosphate discovery is a revolutionary step in that thing. These are all ignored in cancer biology system. Now, the major emphasis on which Warburg made his Arabic glycolysis hypothesis came from the Corey's couple. Corey, husband and wife, they worked in 1920s on carbohydrate metabolism. What they have stated is liver produces glycogen and glycogen secretes actually the sugar into this thing. Sugar will be utilized by the peripheral tissues. They secrete lactic acid into the blood, and that lactic acid will enter into the liver back again, and then lactic acid will be converted to glycogen. But one of the major defects on this particular theory, which later on was recognized by Kaplan and others, was that LDHA, which is LDHB, which is required to convert lactic acid to pyruvic acid, is totally absent in liver. Adult livers do not contain actually this thing. Embryonic livers or carcinogenic livers are different, but adult livers will never actually produce lactic acid. So this glycogen Corey cycle glycogen lactic acid theory was overthrown. Sutherland joined this group somewhere around 1929 or 30. Afterwards, he discovered cyclic AMP. He was awarded Nobel Prize for cyclic AMP as a messenger molecule. And during that period, he has stated that epinephrine, which actually causes actually inflammatory systems, activates inflammatory system. Epinephrine actually contributes to this particular lactic acid production. It is not actually the exercise or actually the glycogen lactic acid theory. It is the epinephrine which hydrolyzes liver production of the glycogen is totally independent of lactic acid, was shown by Sutherland. Okay, so this was amended later in 1940s. These people got Nobel Prize
Sir,
in, I think, yes.
sir, can you wind up by 9:30? It's almost 1 hour.
Okay, oh yeah, that is fine.
Yes, sir.
We, uh,
We'll again
we are
join on when yes, sir, we'll
no.
again join on Wednesday.
Are you getting fatigued? Okay. This is actually the stress factors will cause actually hydrolysis of the muscle glycogen was reported by this thing. One important thing is they shared the Nobel Prize along with another one. This is important. Pay attention. Bernardo House. Okay. He shared 50% of the Nobel Prize along with Corey. Corey's got 25%. This man, what he has stated is pituitary secretions control actually the insulin output by pancreas. Pancreatic secretions are controlled by hypothalamus and pituitary, particularly the adenohypophysis, that is anterior pituitary, controls the secretions, was shown. Even to this day, this discussion, nobody discusses this particular thing, but we have he has also stated that high blood sugar per se did not affect actually pancreatic cells. They are independent of actually this thing. Hyperglycemia may be due to the extra pancreatic factor. And today we have incretins in the form of what is called glucagon-like peptides, insulinotropic peptides like GIP, etc. And these are this thing, and gut hormones play a critical role in insulin secretion, has come to be recognized now. Okay, so this is another missing link in the present models of metabolism. Two important things which we don't understand is first, how is insulin secreted inside the pancreatic cell? Number two, how is insulin functioning in reducing the blood sugar level? Okay, shall we close?
Yes, sir.
Shall we close now, or you have another 10 minutes? You have patience to listen. I'll, yeah, 3 4 slides. I'll explain and close this thing. Now, parallel to Harden, Euler worked on yeast and other metabolic pathways. Yeast differed from Harden on one particular point. He complemented on actually the discovery of thermostable. In fact, Warburg also worked with Euler in the initial stages. What Euler has stated is that now they formulated an equation. Harden along with the Young formulated an equation which is called as Harden and Young equation. You are able to see this? And then Von Euler and his team has formulated a different this thing. There is a variation in this thing. I'll tell you. Harden and Young equation says that two molecules of sugar are reacting with the two molecules of phosphate and reacting with what is called to produce two molecules of alcohol, two molecules of carbon dioxide, two molecules of water, and at the end it produces actually hexose diphosphate. Okay, and this hexose diphosphate in the next stage is broken down to what is called a monophosphate, and then another phosphate will be liberated, was their theory, Harden and Young's theory. Contrary to this, what actually Harden and Euler, this model is more important for our model also. Therefore, I am emphasizing it. I have highlighted it on the yellow line. He has stated that these thermostable cofactors, that the oxidation or transformation of the sugar molecules is controlled by different mechanisms, and oxygen is one of the important mechanisms. Both in the presence of oxygen and absence of oxygen, carbon dioxide is produced. But in the presence of oxygen, he has stated that it activates actually the biogenic reactions. That is, oxygen activates biogenic reactions. It suppresses fermentation, was what was actually shown on that. He did not say suppresses also. He only stated that, when the oxygen is not available, the yeast tissues or other organisms which he has tested, most of his papers are in German, therefore we don't have original. You read his 1929 Nobel lecture, there he explains. So in that actually the fermentation takes place in the absence of oxygen, but when the oxygen is there, oxygen promotes biogenic reaction. His formulation was one molecule of sugar reacts initially with one molecule of phosphate to produce what is called a hexose phosphate. And this hexose phosphate is split into triose phosphate and non-phosphorylated triose sugar. He called it as energy rich and energy poor, phosphate tagged or energy rich. Okay? And this in the second step, what he has stated is the phosphorylated forms condense together to produce hardened ester, that is hexose diphosphate. So there is a substantial variation between these two people, and that is exactly what on re-examining Aldol's reaction, Meyerhoff came up with this particular function. Parallel to this thing, Stokla saw in between 1902 to 1911 or something like that, he has examined actually the human tissues and also animal tissues. He has shown that these tissues, when actually cane sugar is supplied, produce alcohol. Right? This was contested by MacLean, who joined Harden in 1911. But unfortunately, by 1907 itself, Young has clearly demonstrated that fructose and cane sugar contribute to the alcohol production, but not the glucose. Glucose needs to be transformed to the fructose. Is it clear? And that particular thing was missing in this thing. They used dextrose, that is the D form of glucose, for testing this, and they failed to detect this thing. Another important observation which took place in India was sugarcane cultivation is not native to us. It came from Chile, and sugarcane cultivation during the British period was popularized in the last decade of 19th century and then first decade of 20th century. It picked up. And during 1907, the purification of cane sugar into the crystalline sugar, the sugar which you take in your tea and coffee, that purified, refined sugar was, there were few mills around Calcutta which were able to actually purify these things. Although Punjab was actually cultivating these things, and some other states occupied by British people during that period were cultivating sugar, they could only produce jaggery, that is gold, but not the refined sugar. And in those places, including Punjab or other places where refined sugar was not available, there was no symptoms of obesity, no symptoms of diabetes. But even in Calcutta also, because of the price change, rich people whose children were addicted to what is called the sweetness, they developed the obesity symptoms and also developed type 2 diabetes was shown in 1907 itself. Even after 100 years of insulin discovery, we are still struggling to define what is insulin resistance. Fructose is the primary cause of insulin resistance, and over the 100 years of research on fructose, we know that fructose metabolites, whether it is fructose 1 phosphate or fructose 6 phosphate, they will inhibit hexokinase. Hexokinase is important for glucose uptake into the cells, so therefore insulin resistance comes into the picture. Okay, I'll give you this thing. Shall I go for this thing? Okay, 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 or stop.
Yes, sir. Yes, sir.
I am only telling about fructose. What I have told you previously, I have given you the evidence here. This is what
Yes, sir.
actually, this is what actually Euler also stated. One phosphate rich DHAP, another is glyceraldehyde, and there is an enzyme called triose phosphate isomerase which converts DHAP to glyceraldehyde 3 phosphate. GAPD converts actually this thing. This enzyme is also known as actually Warburg enzyme because Warburg and this thing have discovered. In fact, Warburg discovered NADP first in 1935 and NAD years later. But he has discovered only the first step of pentophosphate pathway. The second step he did not discover. That was discovered later by Lippmann. Therefore, he missed the boat properly. Now, what he has stated is, he has actually, this is important to your project also, Jansi, Janvi.
Yes, ma'am. Yes, sir.
This is the phosphoglycerate kinase. This phosphoglycerate kinase is the one which is actually producing ATP in the cytoplasm. Okay? But in the original glycolysis model, only pyruvate kinase was producing two net ATP molecules. Now, of course, after a long debate and nearly 100 years, PGK1 is also shown to produce one ATP, another ATP by pyruvate kinase. Okay? But one important thing here is the 3-phosphoglycerate, which accumulates, is diverted into what is called in biosynthetic pathway during RNA synthesis, during DNA synthesis. And this particular 3-phosphoglycerate is converted to what is called serine glycine biosynthetic pathway, which is called as one carbon metabolic pathway. In fact, it is serine glycine folate pathway, which also produces cysteine biosynthetic pathway. This is more complex pathway. Even I am also investigating some of the unknown concepts now. So this particular pathway is the central area of your research. Is it clear? Okay.
Yes, sir.
I, in my WhatsApp, this thing I have produced. I have given you that particular Celesta Simon's paper on moonlighting enzymes.
Yeah, sir, you have given it.
Under that moonlighting enzyme, along with the 2000 paper of 12 embryo, why in vitro is not same as in vivo.
Yes, sir, you have even given that.
Why in vitro is not in vivo? I have been explaining that they worked on individual enzymes and fluxes, right? So
Yes.
that also I asked you to prepare the table. This also I asked you to prepare the table. But people are only in the habit of actually mentioning the enzymes and delta G values without bothering actually discussing the cofactors. Okay?
Yes,
Entire
sir.
metabolic pathway of glycolysis and Krebs cycle is based on the assumption that NAD and FAD capture hydrogens, transfer it to oxygen. They don't discuss other cofactors like vitamin B components like thiamine pyrophosphate, except in case of pyruvate kinase, that also they discuss it along with the dehydrogenation. Pyruvate, sorry, dehydrogenase in mitochondria along with the dehydrogenase, isn't it? They call it as pyruvate dehydrogenase. They don't call it as pyruvate decarboxylation. Okay, so that actually is the major this thing, and you will know as you progress into the next models. You will be understanding that vitamin B is important both for glycogen hydrolysis. It is also important for converting actually some of these serine glycine biosynthetic pathways, even PSAT, that is serine production through glutamate from pyruvate dehydrogenase. Okay, this sorry, 3-phosphoglyceraldehyde, glycerol dehydrogenase (PGDH). This actually requires actually the uh, paradoxal phosphate, etc. You will be in a position to actually understand that thing when, along with the enzymes, you also mention cofactors. It may not be given even in that article also, but uh, go to the what is called the mechanism of phosphorylase, mechanism of glyceraldehyde 3-phosphate. Any one enzyme, they will give you that the cofactors are involved in that thing, and that will be working on that. So this is the model on the right side. This is actually recycling actually this particular this thing. One important step we need to understand is I have already emphasized EMT, isn't it? Even Pasteur emphasized that yeast undergo modification into globular structure. So epithelia which are delinked from the polarized tissue assume actually the globular structure. They lose actually the membrane, endocytotic vesicles and all other membrane proteins. They become what is called neutral. And one of the important thing that happens is 4F2HC and also the mechanism of what is called endocytosis, which is made by clathrin dependent proteins. Multiple proteins are involved. They will be lost in EMT proteins. So pluripotent stem cells will not be containing that thing. Therefore, nutrient absorption will not be taking place. Only few nutrient transporters which are uh, either conditionally essential or non-essential amino acids will be transported, and that places actually the mesenchymal cells in a proliferative state, keeps them in the metastatic state, but during the oxidation regenerative processes, they need to activate the serine glycine biosynthetic pathway. In normal somatic cells, Parkin will be inhibiting PAGDH and it will not be allowing that thing. And TPG accumulating the PGAM1, which is according to the present models, is converting it to 2PG, is not working as an enzyme. It goes and binds to the second enzyme of the pentose phosphate pathway, will not allow the ribose sugars to be produced. That means pentoses will not be produced after this serine glycine biosynthetic pathway. Okay? And in case of somatic cells, whether it is RNA or in case of proliferative cells, it is DNA, it is P53 that gets activated into this thing. Have you understood? Okay.
Yes, sir.
I'll close with this thing. You understood that PI plays an important role in both inorganic fermentation metabolism and also in oxidative metabolism, right?
Yes,
Here,
sir.
PI is required for phosphorylation. Okay, PI is required for phosphorylation. I have already told you that glycogen metabolism is different from that of lactic acid metabolism. Okay, and that is how actually I placed actually left side the glycogen hydrolysis and the right side lactic acid hydrolysis. I put a blank because this pathway will not exist in the normal cell. It is only lactic acid entering through transporters. There is a molecule called basigen which is attached to this thing. That MCT1 will lactic acid will be entering, and that lactic acid will be in. I think I placed a figure which I have given you the dynamics of lactic acid. We will go to that particular thing. It is not actually the pyruvate that is acting as a reducing agent. It is acting as here. I'll come to this thing. After this, I'll just explain the seventh slide and we'll close it. Okay? Maybe I crossed 10 minutes you have given. Or shall we stop? We'll stop after this slide. What should I do?
Okay, sir. You wanted to continue any slide, sir?
Yeah, actually, because this closes one segment of my lecture.
Okay, so that
That is,
completed, sir.
this is the dynamics of fructose 1,6-bisphosphate phosphorylation and dephosphorylation, which actually activate hexosamine biosynthetic pathway on one hand, and alcohol fermentation and lactic acid production downstream of fructose 1,6-bisphosphate. These two steps, conversion of fructose 6-phosphate to fructose 1,6-bisphosphate and reversal of fructose bisphosphate to fructose 6-phosphate, play a critical role in transformation of fermentation to biosynthetic activity. Have you understood? Because hexose phosphate is channeled into the hexosamine biosynthetic pathway that acts as a cofactor, and that cofactor leads to what is co-transporter that leads to actually activation of some other nutrient assimilations, including glucose into the cells. Is it clear?
Yes, sir.
The section A, this two-step reaction, which is controlled by three enzymes, three types of enzyme. One is activated by hypoxia. Hypoxia is also a stress factor that actually activates a second gene which is known as pyruvate phosphofructokinase 2. In short, it is PFKFB2. This PFKFB2 acts as an allosteric activator of the PFK1. You know allosteric activation, am I right? In enzymology, you have a molecule which binds onto the enzyme that is not actually participating in catalysis, but it is transforming the protein in such a way that protein assumes active state. So that is what is called as an activating allosteric modulator. So here, this particular molecule will allosterically modify PFK1 to activate it so that the flux is maintained and stability of fructose 1,6-bisphosphate is maintained to produce actually the fermentation products. In case of yeast, it is only alcohol, but in case of tissues, it is alcohol and lactic acid. Okay?
Yes, sir.
Fermentation, in case of human tissues, muscle, animal tissues, it is lactic acid and the alcohol. That is what happens in liver. Liver hepatic non-alcoholic fatty liver disease is nothing but in vivo there is a lactic acid production which keeps the tissue in a fermentation state. As long as this alcohol is allowed to be produced, then what happens is there is a dynamic shift between what is called alcohol production, which if it is increasing leads to the fibrosis, and that steatosis protects it from carcinogenesis. But if you inhibit steatosis formation, it leads to actually carcinogenic formation. So hepatic cancers develop when actually ketohexokinases are suppressed. Is it clear? Now,
Sir.
Tigor. Tigor is a tumor protein, TP53 induced protein. TP53 induced glycolysis and apoptotic regulator. Right? This particular protein will be activating another type of proteins under the same gene, but they will be inhibiting PFKFB3, 4, they will be inhibiting PFK1. That means they will be suppressing the action of HIF1 and activating this particular system. Is that clear?
Yes, sir.
Now come to this thing lactate dynamics on the right side. We have seen in one of the slides that fumarate is acting as a terminal electron acceptor during the anaerobic respiration. That means fumarate is also produced in this thing. But how the fumarate is produced requires another half an hour explanation. We do it in the next session. Okay. So here, remember that fumarate is acting as a terminal acceptor in anaerobic metabolism, electron acceptor, given at the bottom. Are you able to see this? Are you able to see the arrow? Are you able to see the arrow? Which I am putting actually. And B, you are getting B, isn't it? The cursor, I am putting the cursor. No, I have a screen, fructose and lactate metabolism, isn't it? Fructose and lactate
Sir, you're not
metabolism.
sharing any screen.
Why not sharing? I have shared, I am sharing the screen. You are able to see the PPT?
No, sir, we can only see you.
Okay. No, again, it's going actually this thing. Okay. You are able to see?
Sir, we can see now.
This is fructose and lactate metabolism. You have seen the previous one, am I right? So let us not get into that thing. You will see that, we have shown that, fructose 1,6 glycogen metabolism is different from that of lactate metabolism. I have already told you on that, isn't it?
Yes, sir.
This slide you have seen, isn't it? This glycogen, this particular step is actually what is actually differing under that. This shuttling between fructose 6 phosphate and fructose 1,6 bisphosphate is important, right? So this I am highlighting here in the second slide to say that, this particular forward direction of fructose 6 phosphate to fructose 1,6 bisphosphate is controlled by two different proteins. One is HIF1, which is a hypoxia inducible factor. Another is TIGAR. TIGAR is a TP53 induced factor that suppresses PFK1, but whereas HIF1 activates PFK1. By suppressing PFK1, what it is doing, TIGAR? It is converting fructose 1,6 bisphosphate to fructose 6 phosphate. Where is it going? It is going to hexosamine biosynthetic pathway, which I have already explained you. Is it clear? Fructose 6 phosphate is converted to hexosamine biosynthetic pathway. I have shown you in earlier slides, am I right?
Yes, sir.
Right? So this coming to the lactate, lactate shuttles, lactate is produced under anaerobic conditions. Okay, from the pyruvate it is produced to lactate. Lactate intracellular it is produced to lactate. Accumulating lactate is exported outside by MCT4 and then that becomes what is called intercellular lactate circulation or blood circulation. But circulating lactate enters into the cells through MCT1. That is, again, contributes to the pool of lactic acid inside the cell. And then this lactic acid intracellular is oxidized by LDHB to produce pyruvate. Pyruvate carboxylation converts it to oxaloacetate and malate. And this actually produces fumarate in a series of steps. I will talk about this. This requires a half an hour lecture. And this fumarate can be oxidized here to succinate. Succinate can act as an epigenetic modifier by binding to the genes, and then it can also activate respiration in mitochondria to produce GTP. Is it clear? Is it clear?
Yes, sir. Yes, sir.
I stop at this level. Whatever PPT I have given, read 2, 3, 4, 5 times till you understand every slide.
Okay, sir.
Do you have any homeworks to do?
Yes, sir. We
Then
don't have.
in that case, you sit 2 hours now, 2 hours, or 10 o'clock. Okay, at least 2 hours in the morning before we come to the end. 2 hours in the night tomorrow. Day after tomorrow, we come over here. Day after tomorrow, I will talk about epithelial mesenchymal transition, how actually epithelial mesenchymal transition in microenvironment reprogram nitrogen metabolism.
Okay, sir.
And Pasteur said nitrogen contributes to growth, right? Euler said nitrogen contributes to growth, oxygen contributes to growth. They need to be integrated, right?
Yes, sir.
Present models say only glucose does everything, it does. Fatty acid will not be produced by this thing. Fatty acids will be produced by the protein digested in the duodenum, which enters into the blood. There it enters into the visceral adiposity. Because of the adipocytes present in the mesentery will translate into the will be converted into the adipocytes. Okay,
Okay,
I
sir.
I close it. Sorry, I took more time than what you, but half an hour we lost it only in arranging your Zoom. Okay. All of you are there or they left?
Everyone are there, sir.
Okay. Janavi, Shreya, you understood that? You understood being a biologist and biochemist, you should be in a position to understand it more. Okay? This, this model is not an examination model, but this model is the basis for research. Okay?
Okay,
Unless
sir.
I told you that we are not in a position to identify proper this thing. If we are in a position to actually put it in the this form, already people are accepting my hypothesis. But there are several questions in the one carbon metabolic pathway. I will address them in the next lecture, maybe after the next lecture. Next lecture, I will converge on fumarate and one carbon metabolism. Okay.
Okay, sir.
I'll be posting some of the papers. Try to read them. Okay.
Okay, sir.
Already I posted them. I posted them. You can just. I have also posted some of the screenshots also. You can just read them. Be thorough with this particular segment. When you come to the next segment, there should not be any opportunity for me to repeat it and take your time. Okay? If you want to save your time and learn more, be thorough with this segment. Thank you very much.
Sure, sir.
Sorry,
Sure.
sorry,
Thank you so
sorry
much,
if
sir.
I have bored you. Sorry if I have bored you.
No,
Thank you.
sir, it's not an issue.
I know it is more than 45 minutes is a boring lecture. Whatever, whoever is the speaker. Okay. Thank you.
Thank you so much, sir.